And next up, we have one of our keynote speakers, Dr. Shila Ghazanfar. So Sheila is an Australian Research Council DECRA Fellow at the University of Sydney. So she did her PhD in Statistics and Statistical Bioinformatics at the University of Sydney before completing a Royal Society Newton International Fellowship at the University of Cambridge. So during her time there, she developed new techniques for the analysis of highly multiplexed RNA fish imaging data, as well as methods for statistical integration with single RNA sequencing datasets. So she recently established her new lab just this month that she'll be focusing on designing new statistical approaches to analyze spatial genomics and single-cell RNA sequencing dataset and different innovative multi-omic data types. So welcome to the stage, Sheila.
Hello, everyone. It's so wonderful to be here. I'm extremely happy to be on home soil and speaking to you here in person as well. This is my sort of very first in-person presentation since pre-pandemic, so please excuse any nervousness. So firstly, I'd like to thank the wonderful organisers for the invitation to present. Like Ray mentioned, I've recently started my fellowship in Sydney, extremely recently, and I'll be describing some work that I did during my postdoc with John Marioni in Cambridge, as well as some work that is ongoing as I transition here to Australia. So for some biological motivation, one of the key interests in the John Marioni lab is the study of early embryonic development in the mouse. So previously, the lab has generated massive transcriptomic time course atlases using single-cell RNA sequencing. So we can see that as embryonic days progress, from the stages of gastrulation into early organogenesis, that there's a huge diversification of the cell types that emerge over time. And so we can see that there's massive shifts happening. However, the key component here is that we're not necessarily capturing the cells in their native tissue contexts. So the sort of place where I came in was looking at how we could incorporate the spatial component into our understanding of early organogenesis in the mouse. And so with a team of some really wonderful collaborators, and I will single out Tim Lohoff, a now graduated PhD student who we worked very closely with, as well as our collaborators Long Kai in Caltech. We performed a spatial profiling of whole embryo sections for a set of hundreds of specially selected genes. And the idea there was to try to understand the interplays of local spatial contexts and how that relates to our understanding of cell fate determination. And so what I'm showing here is a digital in situ of one of the samples that we collected. And so what we can see is that there's a massive shift in the scales of data of the information that we're able to capture. So, we're looking at firstly the whole organism scale into the tissue scale and then further into subcellular. So this technology that we use to capture molecule resolved spatial profiling was seq-FISH, so sequential fluorescence in situ hybridization. And so George did a very wonderful job at explaining the overall technique for how barcoding based image imaging RNA fish works. So for this project, we used this technique on 3 sagittal sections of mouse embryos, and we subjected these samples to seq fish as well as some additional rounds of single molecule fish. Following that, we performed image processing, including cell segmentation and quantifying the molecules in space, and then downstream analysis. So the approach for seq fish is a barcoding technique where a number of individual rounds of images are taken that essentially capture pseudocolors, which are then building up a barcoding scheme for multiple genes. So we were able to use this scheme to capture about 350 specially selected genes. One additional component to this analysis was an approach to capture cell segmentation of our tissues. So in the stages of embryogenesis, the cells are extremely tightly packed, and so we suspected that there'd be a problem of cell segmentation if we were not able to capture the boundaries of cells. And so the way in which we did this was to target a set of proteins that are sort of known to be present on the boundaries of cells, so a set of cadherins. And these proteins were then targeted, and then through a set of through a tertiary probe that then bound to the hydrogel itself, it meant that we were able to preserve the protein signal for the boundaries even after the tissue clearing step for the seq fish imaging. And so what it meant there is that we had one imaging round that corresponded to cell membrane as well. And that meant that we were able to use a random forest-based segmentation approach, elastic, to perform segmentation of cells, not just nuclei. So on to the sort of questions about data analysis. I mean, we had a sort of single-cell RNA-seq reference atlas that we could use. And so what a key sort of question that we had was, we'd like to use all of this curated information to be able to accurately identify the cell types that we see in our spatially resolved data set. And so in the top, we've got our UMAP from our transcriptomic atlas. In the bottom, we have our sort of UMAP that we get from just taking the gene expression from the seq fish data. And our approach here was to perform data integration using mutual nearest neighbors. And the sort of key thing that we needed to do for this was to restrict to the overlapping genes. And so what we can see in this sort of joint UMAP is that we have some quite nice overlap between the two data sets, and we would kind of hope so because they do come from the same types of tissues. But importantly, what we could do following on from that is employ a classification approach to then sort of, well, on the top we just preserve the same labels as what we had before, but on the bottom we were able to classify our spatially resolved cells into these sort of very sort of well curated and in the same vocabulary, I guess, if you would, types of cells. And so what that meant for us is that, you know, we kind of, our spatial image here, and what I'm showing is the segmentations of each cell. What it meant is that we could sort of take this single-cell RNA-seq and effectively paint on the colors for what we have from our atlas. And so here what we can capture is that, we are seeing some very, I guess what we would expect. The dark green corresponds to the, if I've got my cursor, can you see here? Yeah, corresponds to the region corresponding to the forebrain, midbrain, and hindbrain of the developing mouse. This sort of dark mauve area corresponds to the developing heart. The pink is the developing gut tube, and then the surrounding areas. So one of the sort of immediate observations that we made was that cell types are very differently distributed in space. So maybe that wasn't so surprising. But what we can sort of see is that there are many different sort of patterns in which we're seeing cells, especially those that are very restricted and those that are very sort of very dispersed. And what we could do is try to get a handle on how much we could explain the transcriptional variation by the spatial location in itself. And so here we used a linear modeling approach to identify for which cell types could we sort of best predict the gene expression using the neighboring environment. And so these plots here are showing that we found that the sort of highest amount of variation could be explained in the forebrain, midbrain, hindbrain cluster. Now, the fact that it's called the forebrain, midbrain, hindbrain cluster kind of gives us a hint that maybe we could actually increase the resolution for which we're looking at this group. And that was something that we were able to do as a mixture of our curated single-cell RNA-seq data, but then also using the spatial coordinates. So we performed additional clustering for this particular group, and indeed we found that we could break down this sort of three-part group into the forebrain, the midbrain, and the hindbrain, sort of quite together. So that's kind of nice. It gives us a way of sort of seeing that we can use the single-cell RNA-seq and the imaging, the spatial data to try to inform one another. Now, another component that we looked at sort of very specifically was about this question about diffusion in space. So we focused on an area called the midbrain-hindbrain boundary. And so this is our image from our second embryo. And I'm zooming in on this region that corresponds to the MHB. And so we can see that there's sort of a bit of a difference between the annotated cell types, the sub-cell types. And what we could do was take those cells, using the gene expression information, we could embed them into a diffusion component space. And so here we've colored based on diffusion component 1. And if we port over those diffusion component colors, what we see is that there's what we observed was a sort of extremely smooth shift in colors. So I'm overlaying the sort of estimated vector field here. So this is not RNA velocity. It is just the vector field showing the difference in the shift in diffusion component 1 over in space. And so, of course, diffusion components come from many, many different genes. And what we could do following on from that was use a computational technique that I developed sort of at the beginning of my postdoc called SHOT, so single cell higher order testing. And what what this is a non-parametric test for differences in gene expression variation, or gene expression correlation across a trajectory or across a space in general. And so what we found was that we could identify particular genes that have these sort of potentially non-linear patterns that explain different regions of expression. So, we can note that there's sort of EN1 that is present sort of very much along the boundary, and then, say, for example, Has5. This is in sort of our inferred gene expression based on our single-cell RNA-seq, sort of radiating out from what's out from the MHB. So that's sort of one component that we looked at in detail. So this was the brain sort of story, I guess, from this project. Now, another thing is that, we looked at the section of the entire embryo, and what was important was to ensure that this data is sort of able to be investigated and hopefully interactively. So I developed a web platform to have a look at the spatial mouse atlas. So, of course, this has visualization. Remember, we captured expression at individual molecule level, so we can perform sort of multicolor digital in situs with any combination of genes among the library. Another that I'll show a little bit more detail is this question of virtual dissection and differential expression analysis. So hopefully I have this going. So this is a screen video of the Shiny app, and so what is quite nice about looking at spatially resolved data is that you can sort of select and virtually dissect different areas and look at them in detail. And so here I've selected sort of two regions corresponding to groups A and B, and using just a lasso tool, and then from that I can see what is the makeup of the cell types, as well as what differences in gene expression we see across these regions. So that's been quite helpful, and I think that is quite a useful way of being able to just sort of very quickly check things. So moving on from this, I want to sort of just in the last 5-ish minutes kind of switch into these questions of further data integration with spatial genomics. And one project that a very talented master's student who came over from Zurich and did his project with me was to look at this question of spatial reconstruction of single-cell RNA-seq data. So remember, we looked at our spatially resolved data, and we use single-cell RNA-seq to try to help us annotate the cells. But now we have a spatially resolved resource. Could we use this to help us understand where the latent cell positions of another dissociated data could be? And that's the sort of question that we're asking. We don't want to go one way, we want to go the other way. And the approach that Elias developed uses two sort of key components here. The first is a gene interaction network, and the other is a graph neural network. And so the idea here is that if I have a spatial reference, so I have the expression and the physical coordinates, I could then train a graph neural network to then identify which patterns of genes best recapitulate the spatial coordinates. And additionally, I can look at the model itself to understand which genes are best predicting these regions. So that's the model. And the second component of that is, well, if I have some dissociated data, then I could pass it through the graph neural network to then predict the relative similarities in this predicted space. So I can infer a cell-cell distance matrix. And so if we go back to our sort of mouse example, we have our expression plot, but then what we get from SageNet here is a slightly different picture. And so one thing that's immediately clear is that in the expression UMAP, we have the dark green of the brain sort of very clearly being split out into those forebrain, midbrain, and hindbrain components. And yeah, I think that's quite nice. Now, another aspect that I think is sort of becoming a little bit more clear as we have, spatial genomics, single-cell multi-omics, spatial multi-omics, and so on, is that when we're performing data integration, I would say that more and more we're wanting to perform something that's sort of termed mosaic data integration. And so what this sort of schematic is kind of trying to capture is that for various sort of combinations of data sets, we may be capturing some assays for some sets of cells, and we may be capturing others for those. Now, when I performed the integration of the single-cell RNA-seq with the seqfish data, what I did was restrict down to only the overlapping genes. Wouldn't it be nice if I didn't have to do that? And so that's where the idea behind StabMap was born. So this is a stabilized mapping approach that essentially takes in a set of data matrices, so long as there is some sort of overlap among the entire set, where you can infer a mosaic data topology, so you can get a sort of connected network here. StabMap will generate a common embedding space. And the way it does this is by constructing shortest paths along these data sets and iteratively projecting, predicting, and then projecting the cells over. So, we have the data set, say, from this middle data set being projected onto the coordinates of the sort of red principal component or linear discriminant space. And then what happens for, say, another data set is that it will be iteratively projected onto the coordinates of our sort of bridge data set, if you will, and then furthermore into the reference. So this is on bioRxiv, and I just want to highlight just one proof of principle that we did for this, where we took experimental perturbation cells. So these are cells that were, captured using single-cell RNA-seq, and we used the genes that were not in the seqfish library, as well as the neighboring expression from our seqfish to effectively stabilize our joint embedding into space. And so we find that we were able to sort of get a very fine-grained mapping into a joint space. And then, this is experimental perturbation, so we could transfer over the information from the perturbation to then infer the physical regions where we see relative enrichment or depletion of knockout cells in space. And so this gives us a sort of way to design more validation experiments. So to summarize, I've described to you a spatial reference map for mouse organogenesis. We've integrated these data using existing dissociated data sets. We looked at different sort of bits of biology, and I focused particularly on the brain this time. And yeah, so I'm very much looking forward to questions. I'm just starting in Sydney, so looking for a PhD student and for collaborations. And finally, I'd like to acknowledge all of the wonderful contributors to this project. It's been really fantastic. Thank you.
Thank you, Sheila, for a really wonderful talk, really amazing data set that you generated. Do we have any questions from the floor?
Hey, Sheila.
Hi.
Thank you. Fantastic talk. With integration methods, unfortunately, people often sort of treat them as a bit of a black box. They just put their data in and they take whatever method or whatever results they get out. What sort of diagnostics do you look at to assess whether an integration method is doing a good job versus forcing together things that shouldn't be?
Yeah, that's a good question, Pete. I'd say that, I think a lot of data integration techniques these days, they work a lot on this sort of already reduced dimension space. And so then you're putting like PCs of one with the PCs of another. It is useful to just go back to the gene expression and look at marker gene expression. The other is, how much relative enrichment and depletion do you see of different sort of predicted cell types, I guess. So, if you know something about your sample, like for example, we know that our mouse atlas, these were sagittal sections, so we should be seeing maybe a depletion of anything along the left-right axis. That is something that we saw, and so it did sort of give us an indication that it may be that the data integration is not sort of mushing everything together too much. Yeah, so I'd say those are the things.
Cool, thanks very much.
Thanks.
Hi, wonderful talk. I was just wondering, with your single-cell spatial integration, you showed, a label transfer approach and a classification approach, which I wasn't quite clear about how that was based. But did you also do de novo cell type kind of, discovery in the spatial data, and how does that compare to the label transfer approach?
Yeah, that's an excellent question. So in terms of the cell type classification, we used KNN with K equals 5 from within that sort of corrected low dimensional embedding. We did include a sort of stabilizing step to kind of merge the labels of clusters that we saw come together. And then your question about sort of de novo clustering of the spatial data, indeed, we did see, so I think somewhere in supplementary, we have a sort of heat map sort of diagonal of particular groups that come out in terms of the clusters and the predicted and the sort of predicted cell types. One thing that was quite nice is that, like the brain cluster, sort of very nicely we saw that there were like multiple clusters, even though they were all given that same umbrella label. And so that gave us a little bit of a hint, in addition to the transcriptional modeling, that was like, okay, well, we can sort of further subdivide these.
Yeah, that's really nice. I guess one of the things that's so exciting about the high resolution spatial profiling is that you have a chance to see, how an endothelial cell that's located next to, one particular tissue type might be different from another.
Yes.
So that's kind of where I was going, but yeah.
Absolutely, yeah. We have like a little vignette about the mixed mesenchymal mesoderm, about that.
Okay.
Yeah.
Hello, that's a lovely piece of work. Congratulations. And I'm quite interested in your data on the diffusion of those transcripts. I know you mentioned that those arrows are not really indicating the mobility or like the speed of, but I was wondering if you found any correlation between, the expression of, say, a transcript between, because I'm interested in like RNA transport mechanisms before. And so I was wondering, did you find any morphology or functional correlation in a particular transcript on specific locations? Say this transcript is highly expressed on this region of the brain while it's lowly expressed on the other region. And I was like thinking that your work can potentially be expanded if you have a model wherein, say in Parkinson's disease, wherein can you find different expression or this diffusion in those kind of disease settings?
Yeah, absolutely. Yeah, no, I think a sort of maybe glaring hole at the moment is that, yeah, we have the subcellular resolution, but for this particular project, we focused on the cell level. One thing that we did find when we performed a subcellular sort of area enrichment analysis was that, as expected, we found that the transcripts associated with exist, it's a long non-coding RNA that sort of goes towards the DNA. It was close to the DNA as we saw, so that was quite nice. But yeah, absolutely. I think that there's more to be studied in this data set. I absolutely believe that, especially in terms of where particular transcripts are in relation to their neighboring cells or in relation to other transcripts as well. But yeah, it's an excellent question. Thank you.
Thank you, Sheila, once again for a really wonderful talk. And because of the time constraint, we can't really answer all the online questions, but if you type in your questions, we can actually forward those questions to our speakers so then they can get back to you later on.